Welcome to Chapter 13, Part 1. This is basically going to be an overview of the brain and brain structures. We also have Part 2, 3, and 4. I kind of chopped up this chapter because they threw a whole bunch of stuff into it, and I think it makes it a little confusing. So bear with me this time. We're going to have 4 parts. 4 major brain regions. So make sure you're paying attention to these terms like regions or lobes or structures. So cerebrum is the largest. This is what we would point to if somebody said, where's the brain? We'd be touching the cerebrum. So there's 2 hemispheres and there's 5 lobes per hemisphere. So basically if you divide the brain up, you have the left side, you have the right side, and then we have specific lobes, and you'll notice that they're named after bone names as well, or I guess the bones were on top of the brain, so they're named after the brain. But we have frontal and occipital. Diencephalon basically is the midsection, and these will become more clear as we look at the image. So the diencephalon is going to be the thalamus, the hypothalamus, the epithalamus. The brainstem goes from the diencephalon down to meet the spinal cord. And then we have the cerebellum, which is going to be on the posterior or backside, kind of posterior and inferior to the cerebrum. We have this term gyri or gyrus. Gyrus is one, gyri is many. And we have sulci, many, or sulcus. And then we have this term fissure. Fissure is deep, sulcus is shallow. So maybe S and S will help you remember that. Here we have a left lateral view of a human brain. We can see this really large structure is the cerebrum. We can see that there are several lobes here, occipital lobe, frontal lobe, parietal lobe. We can see kind of inferior and posterior is the cerebellum. And then this entire section is the brainstem, which is made up of the midbrain, which you can't see, the pons, the medulla, and it meets the spinal cord. So again, remember right here is where you'd have your vertebrae and you'd have your occipital foramen. Here we have an inferior view of the human brain. Again, you can see the cerebrum, you can see the cerebellum, you can see the brainstem. We'll talk more about those. And then all of these yellow, these are cranial nerves. So we'll talk about cranial nerves when we get to part 4, but they're extending from the cranium to the body, to the peripheral nervous system, and they're traveling through those transverse foramen, those extra holes that are in the cervical vertebrae. This is probably the best view to start with. You can see all the internal structures. So we're looking at a midsagittal view. All of this structure here is the cerebrum. Back here we have the cerebellum. You can see the brain stem. We have here the midbrain. Then we have the pons. Then we have the medulla oblongata. Then we get to spinal cord. Some of the inside structures that we'll talk about, there's this large kind of rubber band structure that keeps the 2 halves, the right and the left hemisphere of the cerebrum together, and that's called the corpus callosum. And then if you remember, I said the diencephalon was kind of the middle section of the brain, and it includes the epithalamus, the thalamus, and the hypothalamus. And we'll talk about some of these glands that hang off as well. We have the pineal gland and we have the pituitary gland. A few comments about traumatic brain injuries or TBIs. They can be because of acute brain damage as a result of trauma. So acute means now, right? It's happening right now. But you can have a cumulative effect. So here with concussion, this is the most common type of traumatic brain injury. You can lose consciousness, have a headache, have drowsiness. But let's say you're playing football or you're doing wrestling, right? They've done lots of studies on repeated concussions and how these can change intellect, personality, mood, based on the repeated trauma happening over and over again. So there's a cumulative effect. A contusion is bruising, and while this doesn't sound so bad, with bruising we're going to have inflammation, And with inflammation comes swelling. And if you remember, the brain is encased in this bone, right? All the skeletal bones that we learned about. There's not a lot of place for fluid to go to. So that fluid can build up and put pressure on brain tissue and damage the brain tissue. Here we're looking at a coronal section of the cerebrum. You can see here that there is both gray matter and white matter. The gray matter would be unmyelinated neurons. And you're going to see this term used a lot in nuclei. We're talking about cell bodies. So here, embedded in the white matter, you'll see a collection of cell bodies that are actually gray matter. So they're cerebral nuclei is the term. White matter is going to be myelinated neurons. And we're going to see axons in the white matter, and we call those tracks. So you should be thinking myelinated should be fast, unmyelinated should be slow messages. Another thing to think about, it's kind of hard. If I have, here's this soma, here's the axon, here's the synaptic knobs. If I'm in the central nervous system and I have a collection of cell bodies, I'm going to call that nuclei. If I have a collection of axons, I'm going to call that a track, only if I'm in the central nervous system. If I'm over here in the peripheral nervous system, if I have a collection of axons, I'm going to call it a nerve. If I have a collection of somas, I'm going to call it a ganglion. So these are just some terms to get straight whether we're talking about the peripheral nervous system or the central nervous system. Another image here, we're at a transverse cut at the brainstem area. So again, do you see that we have gray on the outside, then we have white, and then we have additional inner gray matter. So it's kind of a layering system. The gray is always on the outside, then you're going to have white on the inside, and you could have gray matter inside of the white matter. Now we're here at sea level doing a transverse cut at the medulla, so this is part of the brainstem, and you can see that the gray, the outer gray, is gone. Now we have mostly white, and then we have embedded these nuclei, these cell bodies of gray matter. You can see that this looks pretty complex, right? We're going to find out in a little bit that each of these sections have a specific job. As we move further down to this transverse cut here at the spinal cord, you can see that again there's no outer gray. We have lots of white matter, and then we have this butterfly inner gray matter structure. So this is a little bit more simple, right? This is conveying the messages to and from. This is kind of really complex processing of messages. Next, we're moving on to talk about cranial meninges. So these are 3 connective tissue layers. They're going to help support the brain. They're going to help enclose and protect the blood vessels that are supplying the brain. They're going to help circulate cerebral spinal fluid. So we have the pia mater. This is the deepest. This is touching the brain. Then we have the arachnoid mater, and there's also arachnoid space, and that's where you're going to see lots of cerebral spinal fluid. And then dura mater is a really tough connective tissue outer covering. First we have the pia mater. The pia mater is supposed to be this kind of light pink structure, and you can see it's kind of following the sulci and the fissures, right? Fissure is deep and sulci are shallow. This pia mater is made up of areolar connective tissue, and it's basically like clear saran wrap. It's hugging the cerebrum, so it's hard to separate the two. Then we have the arachnoid mater. This has lots of collagen. You're also going to have space here. You're going to have subarachnoid space, and that's going to be filled with cerebral spinal fluid, which is going to help kind of clean the brain, get rid of waste, bring nutrients, and we'll talk about that coming up in just a second. And then the dura mater is this two-layered system. It's really thick, hard to break, irregular connective tissue. So the whole job of this is to protect the brain and to make room for arteries and veins and to make room for the cerebral spinal fluid to help clean the brain. Another clinical view for you, meningitis, encephalitis. So the meningitis is the inflammation of the meninges, usually caused by some sort of virus or bacteria. If you just want to say pathogen to encompass everything, that's fine. Symptoms, fever, headache, vomiting, stiff neck, may result in brain damage or death if left untreated. So imagine if you have inflammation and swelling, it could put pressure on brain tissue. Brain tissue cannot regenerate. This particular meningitis could be affecting the area of your brain that controls breathing. So that would be a serious problem, right? Encephalitis, this is inflammation of the brain in general, usually due to some sort of viral infection. So this is not just in the meninges, in one of the connective tissue layers, this is actual inflammation of brain tissue. We'll look at an image in just a second, but the brain has 4 ventricles. So these are cavities within the brain. They're lined with those ependymal cells. Remember we said those ependymal cells help create cerebral spinal fluid? Remember they were those cube-shaped and they had kind of little cilia hanging off of them, right? And then these all connect with each other. So all 4 ventricles connect with each other and then drain towards the spinal cord's central canal. Nice image of the ventricles. We can see there's 2 lateral ventricles, one in each hemisphere of the cerebrum. Then we have the 3rd ventricle, which is around the diencephalon, the thalamus, epithalamus, hypothalamus. And then we have the 4th ventricle here, and that 4th ventricle is between the pons and the cerebellum here. And you can see that it continues down to the central canal of the spinal cord. So what does cerebral spinal fluid do for us? Well, 1, it gives the brain buoyancy. If you've ever been in a pool and you feel kind of lifted by the water, you're not as heavy, that's what's happening in the brain. Remember, we have this giant cerebrum and this itty bitty brainstem, right? So we're trying to help kind of support the weight of the brain so that it doesn't crush the brainstem. 2, it offers protection. It's kind of a liquid cushion. It kind of dissipates if you have some sort of trauma. It stops the focus of that energy. It spreads it out. And then 3, environmental stability. So what they're trying to say really is we're transporting away wastes and bringing in nutrients. So think about the fact that your brain has millions of neurons and they're all firing and releasing neurotransmitters. We have to get rid of that waste somehow. We're kind of washing it off. So kind of think of cerebral spinal fluid as a cross between a bathtub and a shower, right? Bathtub because you're floating in it, it feels good. And then a shower because it's really washing off. You're not sitting, your brain's not sitting in its own dirty water. We're kind of washing off all of the waste from all the brain activity that's going on. Another image to show you the flow of cerebral spinal fluid. So the creation starts right here. The choroid plexus within the lateral ventricles. So the choroid plexus is capillaries. So we're pulling blood plasma. And blood plasma has a few electrolytes in it as well as mostly water, right? Then we have those ependymal cells. And those ependymal cells are taking the water and secreting into the watery substance electrolytes like sodium and chloride. And that creates the cerebral spinal fluid. And you can follow the little arrows. It would travel the ventricles into the 3rd ventricle. It could come down here to the 4th ventricle. It can also come back around the complete outside between the brain and the meningeal layers. And then what's going to happen is, as this cerebral spinal fluid is being continuously made, it has to be drained somewhere. So some of it is going to go down the central canal of the spinal cord, but if we have excess, it can flow into the veins, specifically the arachnoid villi, and then into the venous sinus drainage. We do have something called the blood-brain barrier. So the blood-brain barrier is set up from, remember the astrocytes in the central nervous system? Remember the little perivascular feet, and we said these are kind of like the gate guard that decides what can get in and out of the blood vessel. So in the brain, you have astrocytes with lots of perivascular feet, and you have super tight junctions. If you remember when we talked about tight junctions, we said we have those like in the stomach lining so that stomach acid can't get out into the abdominal cavity. Well, in blood vessels of the brain, we have these super tight junctions. So for the most part, let's say 99% of the time, we do not want chemicals to be able to make it from our blood into our brain, right? Think about virus, pathogens, bacterial, anything that's trying to harm our supercomputer brain, right? But some chemicals can get through. So alcohol, nicotine, caffeine, and we do have areas of the brain that we'll talk about that have to release hormones. So hypothalamus has to be able to release hormones. You have your pineal or pineal gland that has to release hormones. So what I'm trying to say is this is a very delicate balance between allowing the items and chemicals that the brain is creating to be released and balancing that with not letting everything get through so that we don't have some sort of infection or swelling or inflammation in the brain. So it's a give and take situation. Now we move on to talk about specific sections of the brain. In part 1, we're going to cover cerebrum, and then we'll cover the rest in part 2 and 3. So this is the most complex. This is where you have all your intellectual functions. So center of intelligence, reasoning, thought, memory, judgment. When I say voluntary motor control, hopefully we're thinking skeletal. So this is who you are, right? Your subconscious mind, your likes, your dislikes, your personality is embedded here in your cerebrum. Here we have left and right cerebral hemispheres, and they're divided by this longitudinal fissure. If I was to look deep into the longitudinal fissure, I would see the corpus callosum. That's the rubber band structure that keeps the two halves together. I'd also like to point out this sulcus. So we have a central sulcus, and it's basically a division line. So if I'm coming from the spinal cord, I'm delivering sensory information to the posterior portion of the brain. If I'm sending out motor information, it's coming from the anterior portion of the brain. And this central sulcus is that division line. And we'll talk a little bit later about precentral and postcentral gyrus. These ridges have specific functions that we'll talk about. The other thing to note is that there's a lot of crossover. So something could be happening on the right side of the body. You could be having some sort of motor issue. If we were to follow the nerve all the way to the brain, it might be originating or located in the left hemisphere. So what they're trying to say is there is crossover. The other thing to note is that people will say the left has certain attributes and the right has certain attributes. They'll say, are you left-brained or right-brained? You're using all of your brain, right? It's not like one whole side of your brain is not working. It's just, do you have more developed areas? Are you more science and mathematical? That would be left side. Or are you more artistic and creative, free thinker? That would be more of a right side feature. But there's been some debate on whether that's really true or not, right? The 5 lobes are the last items that we're going to look at in this part 1. We have a frontal, parietal, temporal, occipital. They're a little bit different in location than the bones. So it's not an exact matchup, but the terms are the same. And then there's an insular lobe, or insula, sometimes you'll see it without the R, and it's not visible at the surface. So the frontal lobe, basically it has a variety of function. We're talking motor control, concentration, personality, communication, decision making. So the frontal lobe, I really like to think of as the action part of the brain, right? The motor part of the brain. The parietal lobe serves for general sensory functions. So think of parietal as sensory. Temporal also has sensory functions, but it's really important in hearing and smell. So all of the messages about hearing and about olfaction or smell are going to go to the temporal lobe to be analyzed. The occipital lobe functions in vision and visual memories. And then the insula, this functions in memory and the sense of taste. So this is again also having some sort of sensory function. I really like this image from another book because it shows you everything kind of colored. So here is the frontal lobe, here is the parietal, here's the occipital, here's the temporal. And then if we pull back the frontal and the temporal, we can see the insula. I think you should also notice here's that central sulcus again, right? There's the division line. Do you see how everything anterior to the central sulcus is going to be motor control? And that's what we just said about the frontal lobe, right? We said parietal was just kind of general sensory information. Occipital was vision, which is still sensory. Temporal, hearing. Insula, taste. So do we see how the majority of the brain here is serving for to receive sensory messages, and then just the frontal part here is what's initiating those motor messages out. And you can also see what I meant by different than bones. This parietal section is much smaller than the actual bone that we call parietal bone, but they're pretty close. All right, I'll see you guys in part 2. Bye.